Welcome back, friends. In this video tutorial we'll be talking about the gram-positive rods. We have already seen the effect of gram-positive coccus like streptococcus and staphylococcus. Now in this case of gram-positive rods, again they are also part of free living bacteria. So let me write, they are also part of free living kind of bacteria, and obviously these gram-positive rods are showing the catalase positive tests. Okay. Now, among the gram-positive rods, there are several different kinds of bacteria we want to talk about, and among all of them, we'll be especially talking about three which are clinically significant, because in this whole series of videos, we'll be looking for those microorganisms only which are clinically significant causing diseases, because we are much more talking about them because we need to control the diseases, right? So, among these gram-positive rods, if we divide, we can take the first member of this kind is Corynebacterium diphtheri or Corynebacterium. So let me write Corynebacterium. Corynebacterium, and the species example is Corynebacterium diphtheri. Okay, so let me write in this species in different color. It is diphtheri. So this Corynebacterium diphtheri is a causative agent of diphtheria. Okay, it is the causative agent of diphtheria. Okay, now the second example for that, the second example of this group can be a bacillus. Can be bacillus. We know that we are talking about gram-positive rods, right? So rod means, definitely I must have discussed this earlier. Rod means we are looking at something which looks something like this. So bacteria will look like this. It may have flagella like this. It can have pili like coming out from outside of the cell membrane, but somewhat it is having the structure like a rod, small rod shaped structure. Sometimes it is having a slide rod structure like that, but still this is not spherical. Instead this is a rod shape, right? So bacillus is a typical example for this kind of structures. And in the bacillus, we are having many different species. Now, especially the clinical significant species that can cause diseases in human are Bacillus anthracis as well as Bacillus cereus. So let me write the examples. Examples are Bacillus anthracis, and Bacillus cereus. So these are the examples of bacillus causing agent, or sorry, disease causing agent of bacillus. Now the third type are listeria. They are unique type of microorganisms because they can transfer from one cell to another cell without being exposed to the extracellular matrix. So they can simply transfer intracellularly from one cell to another cell inside the host. That is the unique ability of them. We'll be talking about this listeria also. The example of listeria that can cause disease is Listeria monocytogenes. So let me write monocytogenes. GENES, monocytogenes. So Listeria monocytogenes will be another important example. So these are the major three types of gram-positive rods that we are going to talk about in this course. Except for that, we are also having different kinds like, let me write here, like Propionibacterium. So let me write Propionibacterium. Propionibacterium is an example for that. There shouldn't be any gap between propionine and bacterium. They are all together. So propionibacterium, and obviously lactobacillus is another example. Why we are not talking about lactobacillus? Because lactobacillus, more strains of lactobacillus are beneficial for us. So lactobacillus, example of the strain will be Lactobacillus lactis, for example. So let me read Lactobacillus lactis and all these things. So these lactobacillus are usually they are beneficial strain. Beneficial. Now, as they are beneficial, we are not going to talk about them because we are going to talk about this lactobacillus species and all these things in industrial microbiology perspective, because they are important for industrial microbiology to use them in different industrial products, right? But not in this case, because they are controlling, they're helping us to low down some other opportunistic pathogen inside our body, so they are good. Okay, and so these are the major kinds of gram-positive rods, and among them we'll be talking about Corynebacterium diphtheri, this particular species, Bacillus anthracis, and Listeria monocytogenes. So these are the three majors. So let me talk about it. Corynebacterium diphtheri, Bacillus anthracis, and Listeria monocytogenes. These three different threatful microorganism. Okay, so in the future video we'll be talking about them. Now in all of these gram-positive rods, they are this rod shaped, and more majorly many of them can grow. So let me write can grow on blood agar. They can easily grow on blood agar. So you can separate and get the colonies on blood agar. Okay, so these are the general properties, and we'll be studying about each of them. Thank you. So far we have talked about the introductory part about the Staphylococcus species. Now in this video especially we'll be talking about the infection. We'll be talking about the infection, or the mode of infection, and what kind of diseases the Staphylococcus aureus actually cause. Okay, so the infections that Staphylococcus aureus can cause are majorly of actually two different types. Majorly of two different types. One is simply called as infection. So let me write it here. One is simply called as infection. So this is the first kind, simple termed as infection. Okay, so this is the first kind, infection. Now infection means if we are having Staphylococcus aureus there, so let's say one Staphylococcus aureus, it enters into our body. So let us provide the body system here. So let's say this is the whole body of ours. So let's do this. So let's say this is the body. So this is the body. Okay, and the Staphylococcus aureus enters into our body, and then what it will do, it will colonize inside our body. So inside our body, what it can do, it can colonize. So from one, it can produce many more Staphylococcus aureus like that. Now this particular phenomena is called the infection phenomenon. So in this case, this one Staphylococcus aureus invading inside our body, and it is making this colonization. So let me write it here. It is called as colonization. Now this colonization will result in the different damage of our host cell tissue and all these things, right? So this is a kind of problem that can be associated with the Staphylococcus. Usually we can see this kind of problem regarding our infection in our skin, most of the cases, but the infection in GI tract and other regions are caused by some other mode of the Staphylococcus activity, and this is the second mode of Staphylococcus activity, and it is called, so let me write the second mode, it is called the intoxication. Intoxication. Now in this intoxication is different than the infection. It is largely and solely caused by the toxin released by the Staphylococcus aureus. We all know, we have already talked about that the Staphylococcus aureus can excrete some toxin, which is called the exotoxin. So let's say if this is the Staphylococcus and it can release the toxin outside. So let us talk about the toxin as this red thing. So this is the toxin that are generated by this Staphylococcus. Now as this toxin is generated by the Staphylococcus here, so these are the exotoxins. So exotoxins produced by Staphylococcus aureus, for example. Now this exotoxin can enter into our body. Now if this toxin enters into our body, then in those cases this toxin can cause diseases or different kinds of symptoms, like for example, so if I drag this different reasons, what are the different symptoms. It can be of severe tissue damage. Tissue damage can be a part of that. Okay, and also different kind of problems, different kinds of what you can say, majorly it is associated with tissue damage, except for that immune system destruction. So immune system problems. So these are the different types of problems associated with this kind of intoxication by Staphylococcus. Okay, and this is solely due to the presence of exotoxin. In the previous case, the infection is solely due to this colonization side, right? So these are the things that you have already studied. Now the third kind of infection that it can also provide, and this is due to the combinatory action of both infection and intoxication. So in this case we will be having infection plus intoxication all together. Both of them will act on the same time inside the host. So we are having this, say this is the Staphylococcus species. So we have Staphylococcus and it enters into the body. So let me draw the body again in this case. So let's say here this is the healthy body. Now when it enters into this body, so they start to colonize inside the body. So as they are starting to colonize, so they'll start colonizing there, as well as they are secreting that exotoxin. So all these things are going on at the same time. They're secreting those exotoxins. Now this exotoxin will further lead effects like immune system problems and tissue damage, right? So these are the three different modes of infection that are caused by the Staphylococcus species. Now let us talk about the different diseases that are generally caused by Staphylococcus. And the diseases caused by Staphylococcus are of different regions. For example, so let me talk about that. For example, let's say the first example we are going to talk about the diseases. The first one is skin problems, skin diseases. Sorry for my handwriting though. The skin diseases that are caused by them. Now what kind of skin diseases that we are talking about? Skin and soft tissue infections can be done. For example, if I look at the different examples for that, and the examples are uh, furuncles, for example. So furuncles are the examples, and also cellulitis. Cellulitis is a type of example, and obviously there are different wound infection like traumatic or surgical infections must be associated. So this kind of infections actually occurred by Staphylococcus epidermidis strain. Remember, epidermidis strain, because the Staphylococcus epidermidis strain usually present in our skin. So they present, so it's a natural flora of our skin, right? You know that it's a natural flora of our skin. So in normal times they won't produce any harm, but if there is any wound, through the wound they can infect. So through wound they can actually infection can be caused, right? So that's the basic goal of the skin diseases. Now the second type of diseases associated with all these things, the second diseases associated are or you can say muscular diseases actually. So musculoskeletal diseases actually. The example of this muscle diseases are arthritis and osteomyelitis. So these are the simple examples. Arthritis we know very common. So arthritis can be caused by different types of agents, but this is one of the dangerous agent to cause arthritis. It's a Staphylococcus arthritis, right? So arthritis is a type of condition, and also osteomyelitis. So sorry, let's put them in better form. Yes, this is them. Myelitis, Litis. So osteomyelitis. Okay, the third type of diseases they can cause. The third kind of diseases they can cause are associated with cardiovascular system. So it's a cardiovascular system. Now for this cardiovascular system, the disease they usually cause are endocarditis. This is not that much common, but still they can cause endocarditis. Okay, and again they can cause gastrointestinal tract diseases like renal carbuncles and also lower urinary tract infection. So let me write the last type. The last type we're going to talk about here is, sorry very much, 4. Yeah, now in this case it's a GI infection or GI tract infection. Now in this GI tract infection we can have diseases like, say, let me take this color. It can be lower urinary tract infection, lower urinary tract infection. Okay, and also renal carbuncle. Actually these are not GI tract infection, actually it's a urinogenital tract infection. So I must put here, so it's a correction here, you must put UG or urinogenital tract infections. Okay, because lower urinary tract infection as well as the renal carbuncle, both of them are associated with this urinogenital tract. Okay, so these are the diseases. Except for that, they are causing the toxic shock syndrome. Remember, I've already talked about the toxic shock syndrome, or TSS. This is a very dangerous type of symptoms that are activating the T lymphocyte in our body massively. As a result of that, they will produce more and more T lymphocytes, and those T lymphocytes will start secreting different types of chemical mediators like cytokines. Now those cytokines are going to trouble our body because the cytokines are chemical mediators and they can eventually harm the tissues associated with these regions, right? So this is the kind of dangerous disease. And also scale skin syndrome and food poisoning can be caused by Staphylococcus aureus. So that's a very important point also. I forgot to mention that the food poisoning is associated. So let me write it here. Food poisoning is associated with food poisoning. Food poisoning is associated with Staphylococcus aureus. Largely associated with Staphylococcus aureus. Okay. And this toxic shock syndrome is again another type of dangerous symptoms. So these are the different diseases and infections caused by the mechanisms, as we've already talked about. The mechanisms can be infections only, it can be intoxication only, or it can be combined effect of infection as well as infection. Okay, so that's it. I hope that's helpful. Thank you. Welcome back, friends. In this video we'll be talking about the pathogenesis of Staphylococcus aureus. It's very, very important. We'll be talking about pathogenesis. Pathogenesis of Staphylococcus aureus, not only aureus but Staphylococcus species. Now Staphylococcus species is having several different modes of pathogenesis. Actually, remember when you talked about the basic features of bacterial pathogenesis, we have seen several techniques, several different stages. And what are those stages? Stages are entry to the host cell, then entry to the host body first, then second one is the entry to the host, attachment to the host cell, third one is the propagation through the host cell, then fourth one is the intoxication and killing the cell, and fifth one is releasing and then the immune modification. Now in this case we'll be majorly, whenever we're talking about pathogenesis, we'll be looking two important features, and the first feature, whenever we're talking about, the first feature is to go against. So the feature number one is to fight against immunity or host immune system. So when we're talking about fight against immunity, we're talking about the immunity of host, right? And the second procedure is to so it's a fight against immunity or manage to escape immunity or host immunity. And the second process is to produce or is to destroy the host cell, right? Destroy the host cell. So these are the two different ways of pathogenesis in most of the bacteria that we will be studying. Now in this case, in case of Staphylococcus species, the fight against immunity, or this first mechanism, or to escape the host immunity, there are majorly two different mechanisms are there, or two different mechanisms are available. Now the first mechanism that we are going to talk about here is, sorry, no, don't take this color, let's take a light color here. Let's take, okay, now the first mechanism is via the attachment or surface modifications or the surface molecules. Let me write the surface molecules. Now what are the surface molecules that we are talking about in case of Staphylococcus species? The surface molecules like protein A. So it's called protein A. It's a surface molecule of Staphylococcus species. As well as we are talking about, let's say here, fibronectin binding protein, or FNBP. It is also called, so let me write the full form, fibronectin binding protein. Okay. Now we know that fibronectin is an important component to hold the structures together, to hold the nearby cells together. So it's an intracellular junction binding proteins. Now this fibronectin binding protein is also presented by the surface of Staphylococcus. If I draw the surface of Staphylococcus in this case, so let me draw it here. So if this is the Staphylococcus surface, now the surface of Staphylococcus is carrying two different things. One is the protein A. So let me draw the protein A here. So this is the region for the protein A. So this is the protein A, which is a surface protein, and also it is having fibronectin binding protein. So let me draw the fibronectin binding protein. This is the fibronectin. So these are the two different modifications. This is FNBP and this is the protein A. Okay, so these are the modifications that are present. Now, what is the importance of having or expressing these important proteins onto the surface? First of all, the protein A actually helps. This protein A actually helps this Staphylococcus to escape the immune system, or to escape the engulfment via the different immune system cells like macrophages, right? Because if you recall your understanding of immunology, you know that there are cells like macrophages and also natural killer cells, and all those phagocytic cells, they can come and engulf the foreign particles like this bacteria. So in this case, what they can do, they are expressing this particular thing on their surface. So if we are talking about this particular part, that yes, this is the Staphylococcus, and onto the surface of Staphylococcus they are expressing this particular protein. As a result of the expression of this particular protein A content, this protein A is having a tendency to bind with the Fc portion of IgG. So if this is the Ig molecules, and so let's draw the structure. So this is the IgG molecules. Let me write, this is IgG antibody. Now this is the Fc portion of IgG, right? So let me mark it. So it's a Fc portion of IgG, right? And this is the protein A particle. So this one is the protein A, right? Now what this protein A is doing, the protein A is having the tendency or capability of binding with the Fc portion of IgG. As a result of that, what it is doing, that it is escaping the endocytosis. So as a result of that, so no endocytosis. So let me write, no endocytosis is possible. No endocytosis is possible. Why? Because we actually know that in most of the cases, what is the basic thing, that if this is the bacteria, for the engulfment of this bacteria, it is need to be coated with antibody. So antibody will go and bind like this. So in this binding, what we can see, that this FAB portion is to be attached. FAB means this is the section which is called FAB. In this case, this regions, right? Now FAB is the portion which is need to be attached directly with this antigen, because this FAB is the antigen binding site, right? So if it is coating this antigen or foreign pathogen with this FAB portion, in those cases the macrophage cells or the phagocytic cells can come and engulf it. So let me draw the cell and see the engulfment. So now the macrophages will come and start to engulf this total component. So it's a macrophage, it will start engulfing, right? But in those cases, in this case of this protein A is designed in such a way so that it will bind with this antibody not via the FAB portion but via the FC region. So as a result of that, there is no binding of macrophage. So macrophage won't recognize this antibody bind or antibody coated bacteria due to the opposite kind of binding. Now in these cases, it should be the binding that FAB attached to this surface, but in this case the FC is attached to the surface. As a result, macrophage won't get this coated bacteria, won't get the signal, and it is unable to engulf this kind of bacteria. So this process of escaping the macrophage and other phagocytic cells, and there is no endocytosis. Okay, so this is one kind, and second kind is a fibronectin binding protein, or FNBP, which is also associated with this part. So this is the FNBP, or fibronectin binding protein. As it is binding with the fibronectin binding protein, it can bind with the fibronectin layer. As a result of the binding of itself with the fibronectin layer, it is a kind of stabilized at a particular place, and it is not providing any further signal to the immune system cells, so that they cannot recognize this particular pathogen as foreign. So as it is attached to the fibronectin, it will recognize it as its own self. So so our immune system is getting puzzled, that immune system is not getting the signal that this is a pathogen, right? So it is escaping to provide the signal via this FNBP protein. Okay, so these are the different mechanisms of going against the immunity, right? Now the second thing is the destruction of the host cell. Now the destruction of the host cell is largely caused by exotoxins, right? It is caused by exotoxins. Now the exotoxins, as well as by enzymes, several types of enzymes. So these are the two important mediators. Now the exotoxin that it talks about, majorly they are two different types of toxins, and the toxin is, one is the cellulitic toxin or cytolytic toxins, which will directly rupture the cell, directly rupture the host cell. And second types of toxin is called another kind of immune toxin. So let me distinguish these two parts. So it can further divide into two different. One is the cytolytic toxin. Another one is here is immune toxin. Immune toxin. Okay. So the cytolytic toxin, what cytolactic toxin can do, cytolactic toxin actually, if this is the cell, for example, so let's talk about the red blood cell, for example. Most of the time the cytolytic toxin is associated with the red blood cell, so they are hemorrhagic, right? So if we are talking about this is a red blood cell, so if this is the red blood cell, now due to the activity of this toxin, this red blood cell eventually getting ruptured and all these components will start to come out, right? So this process is achieved due to the cytolytic toxin. Okay, and the immune toxin can bring more and more immune cells, and it will force our immune system to generate more and more chemical mediators like interleukins and cytokines. As a result of that, this will cause the toxic shock syndrome, or TSS. Remember, we have talked about it before. They can cause TSS. Now the immune toxin mode of action is something like that. We know that the T cell, or what you can say T lymphocyte cell, need to be activated, and they are attached with certain kind of antigen presenting cells, right? So there is an interaction between T cell and the antigen presenting cell. So for example, if this is the host cell, so let me draw. If this is the host cell, for example, the host cell is having its own MHC molecule, right? So with the help of its own MHC molecule, it is providing the fragment of the pathogen that enters into the cell, and then T cell will come, and T cell will be attached. So this is the T cell. With the help of its T cell receptor, or TCR, it will attach with this MHC molecule, right? So this is the T cell and this is the host cell, right? Now this interaction is important because this will provide some interleukins as a signal to this T cell, and it will bring more T cells into this place, right? So this is the basic process of action or immune action. But in this case, what they can do is they are having this immunotoxins, or we can also call it as a super antigen, right? We call it a super antigen. So let me provide super antigen. Now this super antigen is again a kind of exotoxin, immune exotoxin, provided by the Staphylococcus species. Now this super antigen, for example, let's say this red thing is a super antigen in this case. Now they provide this super antigen there. Now as they are having this super antigen, providing this super antigen, now the super antigen can go and fit onto this place where they supposed to show the antigen. So instead of this normal antigen, they are now filled with super antigen. Now if this super antigen replaces the normal antigen in this MHC and TCR receptor region, it will activate more and more T cells. So this response of T cell is getting much more amplified in this region, and not only that, it will bring more cytokines in this place, more cytokines in this place. As a result of these chemical mediators, it will lead to certain kind of tissue damage. Certain kind of tissue damage will be associated with these things. Okay, and it will cause some disease or some infection symptoms. So these are the major ways of utilization of the toxins in this case. Now the second thing are the enzymes, right? Now the enzymes provided by the Staphylococcus species are coagulase, catalase. So let me write some of the names. Coagulase, they are catalase. Remember, catalase can break down peroxide into water and oxygen. They can produce massive amount of oxygen from there. Coagulase can actually literally coagulate certain bacterial cells one with another and can actually agglutinate there, right? And obviously they can provide hyaluronidase and fibronolysine. Now this fibrinolysine and hyaluronidase, so let me write fibrinolysine. So fibrinolysine, this means lysine means the lysis, right? So they are associated with the lysis of fibronectin. So they can actually eventually degrade fibronectin. They can degrade the hyaluronic acid barriers. As a result of that, they will degrade, or they will what we can say, degrade the tissue lining or make the tissue lining thin. So tissue lining tissue lining damage or tissue lining damaging is associated with all of these factors. Tissue lining damage is associated, right? So these are by the cause of this kind of enzyme. So when we are talking about enzymes, so let me clear these things for you. These are the enzymes that are associated. Okay, and if we are talking about the another important thing that can be caused that are secreted by this Staphylococcus species are slime production. Now, why slime production is important? Now, the production of slime. So let me write it here. This is another kind. So slime production. Okay, because the slime that they produce, this will help them to escape the phagocytosis. So it's the same kind using like that. So you'll escape the phagocytosis. Actually, for the Staphylococcus epidermidis or epidermitis, they are producing this slime. Okay, so uh, so these are the different modes of their infectivity and their pathogenicity also, and they can escape it via they escape the immune system via protein A and fibronectin binding protein, and they can destroy the host cell via the cytolytic toxins and also super antigen presentation, which is a causative agent of toxic shock syndrome. We have already seen that, and they can produce slime, and they can produce the enzymes like coagulase, catalase, fibrinolysin, as well as the hyaluronidase. Okay, so that's it, and I hope that's helpful. Thank you. So far we have talked about the different pathogenesis schemes of Staphylococcus strains, and obviously we've seen that Staphylococcus can create many variety of diseases or different kinds of symptoms. Now in this video we'll be focusing on how to treat a Staphylococcus infection. Now treating, or let me write, we'll be talking about the treatment. We'll be talking about treatment. But again, treatment of Staphylococcus species is quite difficult because there are many strains of Staphylococcus that give rise to different kind of antibiotic resistance, right? Previously, we treat them using antibiotics because whenever we're talking about the treatment of a bacterial infection, the first thing that comes to our mind is antibiotic, right? So antibiotics. So antibiotic treatment is one of the solutions, but the previously antibiotics that are used in previous times are usually penicillin G, and also they've used methicillin. So let me write methicillin, and also they have used uh, vancomycin. So let me write vancomycin. So they have used all these three major types of antibiotics to kill Staphylococcus bacteria inside our body in different situations. But nowadays, all of these three are what you can say, they are non-reactive because bacteria, the Staphylococcus bacteria, give rise to the resistance against all of these three major type of antibiotics. So now they are penicillin G resistance, methicillin, as well as vancomycin resistance. So there are strains that are resistant to all these three types of antibiotics, and the reason for this antibiotic resistance, so let me write the reasons for this resistance. There are two different reasons. One is the hospital-borne infection, and the second reason is the colony accumulated infection. In both these cases, they have given rise to all these uh, antibiotic resistant strains, and they are no longer uh, killed using penicillin G, methicillin, or vancomycin. Now, usually, very at the very beginning, this kind of what we can say, this kind of antibiotic resistance give rise during the 1970s most of the time. But the last kind of antibiotic that was used to treat this case, like methicillin, and this methicillin was proved to be active until 2000. So it is proven to be active until the year 2000. After that year, after during this 2000s and after the 2000s, this methicillin is found to be inactive in staphylococcal infections. So that is making us very challenging to cure staphylococcus infections. So in those cases, when you are encountered with this staphylococcus bacteria, the major things or the major concerns, so let me write what are the major concerns. So major concerns in this case, you need to take care of yourselves, and those concerns are, those are healthy habit healthy habit, or healthy, let me write HA. So healthy habit is a part of this major concerns, and obviously you need to wash your hands. So washing hand is very important before eating. So you need to maintain all these hygienic steps to finally think, or to finally be sure that the staphylococcus aureus infections is not there in your body. But if it is there, there are other type of treatments. Usually the treatments are devastating, they're dangerous kind of treatments, like making incisions, and obviously cutting that particular incision out, and obviously they take out all the fluid that are there in the skin lesions, right? Because majorly they cause three types of diseases. We know that majorly there are three different types of diseases. One is toxic shock syndrome. So let me write, one is a toxic shock syndrome. Generally this toxic shock syndrome is brought about using those toxins, those super antigens, right? Now the second way, or second kind of disease or disease condition they cause is via, so let me talk about it, it's via skin lesion, or you can call it, it's a kind of skin, or skin, so skin kind of coming out of your hand and all these different regions. So skin coming out. So I'm writing it as I forget the name of the medical term, but skin disease actually. Let me write as a skin problem. And the third kind, the third type of condition they caused is a gastroenteritis. Gastroenteritis. So these are the three major types of concern that are caused by the staphylococcus. And in all these cases, you can see gastroenteritis is directly related with food habit, right? So it is related with food habits. So you need to be very careful choosing your food, and be better for your food hygiene. And for the toxic shock syndrome and as well as skin problems, you need to look for other healthy habits like that. Okay, so these are the different types of treatment that are available nowadays, but you need to be very much careful when you're handling all these different uh, parts. Okay, because we are having resistant strains coming out, right? So these are the major concerns, and I hope that's helpful.